These are embryo models that were made from stem cells. It was grown in our lab without the use of egg, sperm, or uterus. There have been many attempts before to try to grow mouse embryos at advanced stages that they have organs, but mostly really these attempts were failed attempts. And we were the first to really be able to grow a fully organized embryo that has all its organs. It has been a very long process of slowly, gradually being able to grow the embryo for more and more days and at higher efficiencies and higher quality. One of the major challenges that we face in modern society is finding organs for transplantation. It's very hard to find donors, suitable donors, and people die from this. You first have to learn how do you grow a natural embryo outside the uterus, and then use these conditions and take stem cells and put them in these conditions. The embryo, which is the best 3D printer machine basically, because it makes organs in the perfect way. So if we can understand and learn some principles about that, maybe we can go back to the dish and try to mimic and imitate that process. We thought that maybe we should use these kind of neutral conditions and basically ask whether such conditions will allow the stem cells to self-organize and propel themselves rather than us actually dictating to them to move on to the next stage. And very much so, they did do the job. The embryo, it typically has 3 different cell types that make 3 different compartments. One will make the placenta, a second one will make the yolk sac, which is the membrane that surrounds and protects the embryo, and one will make the embryo proper. We're having 3 types of stem cells from each of these compartments. Each one you can see here in a different color, blue, green, and red. And over 6 days, you see how basically they're starting to organize more and more until here in the final day, you can just amazingly see how everything is in the right place. We are very excited now in the lab that we've reached a stage we can extend our studies from mouse to human stem cells. My dream is to truly be able to understand and emulate these first 4 weeks of human embryo development. We are completely in the dark currently about really how does the human embryo makes its organs in these first 4 weeks. I think this holds the key to a lot of solution of a lot of the biomedical problems we have. This is no longer science fiction, but this is rather moving to becoming science.